The human eye is remarkable, for it alone provides us with most of our knowledge of the world about us. Yet human vision is really very limited. We can't, for example, penetrate the shell of an egg to see the growing life inside. Nor are we able to see beyond the limits of our own galaxy with the naked eye. These shining objects are not stars, but microscopic plants. The infinitely small is just as hard for us to see as the infinitely large. From the whirling nebulae to the pulsing life inside an amoeba, there's a hidden world that most of us never see. The secret world of life beneath the surface of the Earth. Or the mysterious life of trout living beneath the mirror-like surface of a stream. If only our unaided eyes could see nature revealed in startling scenes like this. The human eye is capable of many feats, but beyond the range of our eyes is a whole world you never see. Some years ago, a group of filmmakers decided to make such optical fantasies come true. Often they would meet in the local village pub to discuss new photographic techniques.
Do you plan to put a cover over it?
Well, I think, I don't know, if you have a cover, you've got the advantage of being able to possibly film.
Though we are now able to film even more extraordinary sequences, our present achievements would have been impossible without the pioneering skills of filmmakers such as these.
I don't think so. I think they catch
15 to 20.
Bigger animals.
And about at least 6 feet.
Gerald Thompson was the founder of the group. He's a forester by training. He first started to make films to show the way in which insects damage trees.
The road and so on behind that.
Peter Parks is a draftsman and also skilled at design. He's invented new devices to enable microscopic plants and animals to be filmed. John Cook was a curator at the Museum of Natural History in New York. He's a world authority on spiders.
It's going to be a deep water tank because you get a lot of absorption.
Like the rest of the group, John Paling is a graduate from Oxford University. He lectured there on freshwater biology. David Thompson, Gerald's son, specializes in filming small mammals.
You run your tank area.
Sean Morris is the time-lapse expert. All these men are scientists, but scientists who are also superb technicians. Their work takes them all over the world, but most of their revelations are made in their small studio down a leafy lane. The group set up its studios in the village of Longhanborough in Oxfordshire, close to where Winston Churchill was born. It was in these modest surroundings that discoveries were made which would change the way we saw the natural world around us. On a summer's morning, Peter Parks walks through the woods to the studios. Part of the success of the group lies in its ability to look at the commonplace in a different way. Held in the leaf of a teasel, a kind of thistle, is a drop of water, perhaps splashed there by someone walking by. For Peter Parks, the water drop contains a familiar world, but it's one which the rest of us don't normally share. As a zoologist, each member of the group knows how his biological subject will behave in the wild. As technicians, they design apparatus which will allow it to behave naturally in captivity and protect it from harm. This camera bench, designed by Parks and built by him in the group's workshops, enables him to film microscopic objects against a dark background. It would be no good expecting to find life within a drop of sterile tap water. This is a typical drop of pond water, which is populated by animals and plants so minute that thousands at a time can pass through the eye of a needle. These pulsating bean-like creatures are cyprids, crustacea far smaller than the point of a pin. Not an exquisite plant opening, but a colony of single-celled animals called vorticella. A cloud of single-celled animals, paramecium, which are the most common found in stagnant pond water. Each is only a few thousandths of an inch long. Stentor can live in colonies, or if circumstances change, it can break up and become a cloud of free-swimming individuals. The droplet also contains a garden of microscopic plants. Volvox contains both plant and animal cells. It belongs to a primitive group called the flagellates, which all carry a whip-like hair at some point in their life cycle. The spots inside each Volvox are known as daughter colonies. The plant reproduces by ejecting these into the outside world. Inside a drop of water no more than 1/4 of an inch across, quite large animals give birth. This Daphnia, or water flea, rows about using its 2 long antennae. It feeds by filtering algae from the water. The eggs and young are held within a brood pouch and then finally ejected. The teeming microscopic community of the water drop is a world that is only put into proportion by the intrusion of a commonplace object, a pinhead. Life in a drop of fresh water is incredible enough, but what about seawater? Peter Parks' technique can show you an even more bizarre world in the splash of a wave. The planktonic creature seen through the eye of a needle is a copepod. Copepods occur in such vast numbers in the sea that they form the first link in the marine food chain. The same drop lurks the spidery larva of a mantid lobster. It's a fiercely predatory animal with claws for grabbing its even smaller plankton prey. These are aquatic snail larvae. They extract nutrients from the water with a row of waving cilia, or hairs. weird and beautiful life forms, highlighted by dark ground illumination, seem to float through space. But the secrets of aquatic life aren't all microscopic. To study larger life forms, John Paling goes to a trout stream which flows right past the studios. The water may look clear from the vantage point of a bridge, but when you start filming underwater, it's about as clear as vegetable soup. Paling decided to build his own trout stream inside the studio so that he could guarantee crystal clear water. Of course, all the aquatic fauna and flora came straight from the real river. In order to encourage the trout and other fish to behave naturally, he had to get a number of details exactly right. The water had to be at the correct temperature and well aerated with oxygen. Even more important, if Paling wanted his captive trout to spawn, he would have to provide them with a flowing current of water. When they're ready to spawn, trout swim upstream against the current to areas of shallow water where the riverbed is covered with a layer of small pebbles. They behave quite naturally in Paling's artificial river. As the females are ready to spawn, the males fight to get close to her. The female starts to hollow out a shallow nest in the pebbles, using strong beats of her tail to whip up a current. The dominant male has managed to fend off the opposition and is alongside her. He shivers to excite her, and she responds by cutting out the nest further. The male shivers and rubs his flank against the female, and she lowers her anal fin into the nest, feeling exactly where she will deposit her eggs. The male casts his milt over the eggs. As the current gradually disperses it, the eggs only need a couple of seconds to be fertilized. Immediately, the female begins to cover the fertilized eggs. The gravel must be of exactly the right size so that it will protect them and yet allow water and oxygen to circulate. A few are lost to be snapped up by unsuccessful males lurking downstream, just as they would be in a real stream. In the wild, even the hidden eggs would not be entirely free from predation. Little believing it would work, Paling introduced a dipper into his artificial river. Immediately the bird began to dive beneath the water surface to search for trout eggs. Filming nature takes more than knowledge and skill. It requires a great deal of patience. John Paling had to wait for the right time of year and exactly the right weather conditions before he could try to film puffballs in action. Puffballs are a type of fungus, and this particular species grows on rotting tree trunks. They have an ingenious method of distributing their spores. Their method of reproduction gave Paling the idea for an unusual sequence of film. When the leaves are stripped from the trees in early winter, the puffballs wait ready and primed like a miniature minefield. All they need is a rainy day to provide the detonator. On the bare branches above them, enormous drops of rainwater gather. Small raindrops would not be heavy enough to force out the spores through the hole in the puffball. It's not only inanimate raindrops which help the puffball spread its spores. Small mammals, such as this wood mouse, are often unwitting dispersers of fungi. As the mouse is unable to rid itself of all the spores clinging to its fur, they're carried far afield and finally fall off. David Thompson, the small mammal expert, has also used a lot of patience to create a set which will allow him to film the secret life of the mole.
Right, you just sit in there. Okay.
This set has been designed to enable David to film a subterranean creature, the European mole, while it's still apparently moving about underground. The set, an imitation of a mole's run, rotates on an old car axle. When the wheel is turned, the mole starts to crawl forward while staying in the same place exactly in front of the camera. It's an ingenious way to show the mole's subterranean travels when all you can normally see from the surface is the spoil from its excavations. Images of the mole moving about its underground tunnels were used to link together other details of its life, such as the rearing of its young. But the filmmakers were planning even more ambitious attempts to reveal the world you never see. This miniature pond is bathed in cold light. Gerald Thompson, founder of the film group, is an entomologist. Most attempts to film insects used to fail because the warmth generated by the lights would overheat the insects and kill them. Gerald combined filters with a shield of water which absorbed the heat. It also concentrates a soft pool of cold light on the subject and so appears more natural. The subject before Gerald Thompson's lens is a particularly delicate one, an emerging mosquito. The legs of the insect are coiled inside the pupa like a spring. They begin to unwind. The head of the pupa cracks open, ready for the mosquito to exchange a life in water for a life in air. The many-lensed compound eye peers at the surface world for the first time. The insect twitches to free itself. The high-powered camera lenses reveal the extraordinary plastic nature of the water's surface film. It looks almost like molten glass. But once the mosquito is sitting on the surface, the water resembles an ice rink on which the insect slides at the slightest movement of air. Gerald Thompson's earlier scientific career was dedicated largely to studying the insect pests that attack trees. He's particularly interested in a rather insignificant-looking insect which has a strange life cycle. He's trying to film the behavior of the largest species of wood wasp, an insect which specializes in assaulting conifers in a most remarkable way. He positions a length of specially sawn pine before his cold lights. The wood wasp slowly patrols the bark, prospecting for a suitable site in which to lay its eggs. Immediately below the abdomen is the ovipositor sheath. The ovipositor itself, the egg-laying device, is already boring vertically down into the wood. It's a high-powered drill capable of penetrating more than an inch into the tree trunk. The drill is made in 3 sections called stylets, which slide one against the other. The sheath protects the stylets when they're not in use. The ridges of the outer surfaces of the moving stylets do the actual drilling. When the ovipositor is fully home, the eggs are carried down between the stylets to its tip. In the wild, they'd be laid in the narrow tunnel made by the drill. When all the eggs have been laid, the wood wasp withdraws its ovipositor from the tree. Under natural conditions, the eggs are packed deep in the wood. This is how the eggs look when laid in the natural state in a tree trunk. As far as the wood wasp is concerned, the job's finished. Virtually all ichneumon species lay their eggs in or on the grubs or pupae of other insects. You'd think the grub of a wood wasp would be safe, but a female ichneumon can detect where it lies. she begins to drill to reach it. So fine and rapier-like is her ovipositor that at first she keeps it within the sheath to support it while she drills. As her ovipositor sinks deeper into the tree, she suddenly whips the protective sheath away and goes to work solely with a naked blade that bends under the strain. The wood wasp grub stirs. It'll be one of its last movements. Unerringly, the ovipositor reaches it, but it doesn't lay an egg there immediately. She injects a poison that doesn't kill but paralyzes the grub. Then she withdraws her drill for the next act in the drama. She doesn't always make a second drilling, but in this case she decides to bore a fresh hole into the tree trunk to lay her egg. The single egg is long and worm-like. It's laid on top of the dormant grub on which the ichneumon young will feed when it hatches out. Soon she will seek out another wood wasp victim, perhaps in a quite different pine tree. All that remains for the present is to pull out her long ovipositor, sheathe it again, and clean up after her exertions. The ichneumon egg lies coiled on the grub like a time bomb. As an entomologist, Gerald Thompson finds wood wasp and ichneumons exceptionally fascinating. But for sheer beauty in the insect world, he turns to butterflies. To film exotic species, it's usually necessary to grow plants not native to Britain, such as a crop of milkweed from Florida. A group of beautiful Gulf fritillaries feed on the nectar of this plant. The butterflies mate on yet another specially grown plant, passion flower. The first eggs to be laid appear as small yellow dots. You might easily overlook them with the unaided eye, but Thompson's camera reveals them looking like a tiny hand grenade. The life which eventually explodes from this is a minute caterpillar. Gerald Thompson filmed this fritillary caterpillar at the exact moment when it was preparing to turn itself into a chrysalis. First, it spins a pad of web on a passion flower stalk. It will hang from this by a pair of hooks as it goes through the process of internal body changes or metamorphosis. You can see the last skin change to the chrysalis taking place in this highly speeded up sequence. First, the outer skin of the caterpillar shrinks while the future body of the butterfly inside contracts. The old skin of the caterpillar has to be shrugged off towards the top of the slowly forming chrysalis. All this time, the only thing holding it to the stalk are the two hooks in the old caterpillar skin. But that skin is about to be discarded. It's the chrysalis that has to hold on now with its own hooks. Now, greatly speeded up, the final stage of becoming a chrysalis. One chrysalis doesn't succeed and falls to the ground, where its chances of producing a butterfly will be very small. For the lucky ones, the moment of emergence arrives many weeks later. The Gulf fritillary is possibly the most beautiful butterfly from the southern states of America. Now it pumps up the tiny veins in its still soft mother-of-pearl wings. They harden on exposure to air. Sean Morris is a beekeeper. Like many of the group, he sees in his own pursuits novel approaches to nature filming. He's even built a special runway from the garden outside his studio to a honeycomb in front of his cameras. He did this in order to record minute details of the honey-making process. But Sean is the group's time-lapse and high-speed film expert. So it was perhaps inevitable that he should turn his high-speed cameras onto bees. Bees in flight. He discovered, to his surprise, just what inefficient aviators they really are. By exposing one frame of film every quarter of an hour, instead of 25 frames each second, Morris makes fungi burst triumphantly from the ground only to wither, apparently, seconds later. The milkweed plant bursts into a ripeness of seeds. John Cook's challenge was to reveal the secret life of spiders. A tarantula bite, though painful, certainly isn't lethal. Nevertheless, it pays to handle a tarantula with care. Its body and legs are covered in fine hairs which penetrate and irritate the skin of anyone who annoys it. But it isn't necessary to go to tropical lands to find fascinating spider behavior. Far more exciting species than the outsized tarantula live in the green English countryside. On a recent field trip, Gerald Thompson searched for specimens for John Cook's cameras. The species they were looking for was a purse-web spider called Atipus. The spider makes a cigar-shaped web among the grass roots. Atipus lives completely hidden inside this tunnel. Its home looks rather like an animal dropping, where it waits for a victim to step on the outside. The spider's entrance is at the bottom of the web, where it seldom shows itself as openly as this. Atipus relies entirely on touch and vibration to know when an insect has alighted on the outside of its web. It positions itself below and makes a sudden grab through the web. This time the fangs missed. When another fly approaches, the spider is ready and waiting. This time it makes a catch. The fangs inject a poison to kill the victim. But now Atipus has to drag the fly into its lair. First, it allows time for the venom to take effect. When the fly begins to quieten down, Atipus deliberately cuts a slit in its own painstakingly woven web and then pulls the green bottle down into its tunnel. The spider is left with a hole in its roof, so it immediately goes to work to repair the damage with a fresh web covering. One of John Cook's favourite spiders, Segestria, lives in old brickwork. This brick wall is only 2 feet square. It's yet another set in the studios of Oxford Scientific Films. Segestria is sometimes called a fishing line spider. Its web consists of a series of fine lines laid over the brickwork. The spider sits in the middle, feet feeling the lines, just like an angler waiting for a bite. A bee advances until it eventually touches a line. The line gives a telltale pull at the outside of Segestria's lair. The spider waits a second or two to be quite certain its prey is really there. Often the spider darts out to land its catch immediately. The bee tries to sting the spider, but the sting gets caught in the web and is pulled out bodily. The poison sac has muscles of its own, which continue pumping. Now Segestria has only to immobilize the bee and eat it at its leisure. The spider does leave one telltale clue to its activities, which potential victims would do well to spot. The ground beneath is covered in debris. Even spiders find parts of their meals indigestible. The group even managed to invent a technique which allowed us to see through the surface of an egg, to see the new life developing inside. Having sawn round the shell, Peter Parks prizes it open with a scalpel. Keeping it submerged in a special fluid which will permit the early stages of life to develop, he carefully decants the albumin, which contains the as yet invisible life, and the yolk that will support it. By this means, it's possible to film the vital first 3 days of the chick's growth. After that, a second method has to be used to capture the story of the remaining 17 days before the chick emerges. This involves cutting a circular window without damaging the fragile membrane inside. Parks carefully scribes the circular window with a pair of calipers. The yolk provides the fat and protein. The albumin is mainly a source of water. The embryo lies in an amnion, a fluid-filled sac which supports and cushions the developing tissues. The eggshell allows oxygen to diffuse through into an airspace where it's absorbed by a network of capillaries. These spread out over the yolk. At 2 days, the heart is pumping. Individual corpuscles can be seen hurrying through the newly formed blood vessels. The blood carries oxygen to the embryo. Carbon dioxide is eliminated through the eggshell. At 4 days, the embryo, drawing its food from the yolk, is already developing a network of veins and arteries. Both brain and eye can be seen at 5 days. The chick stirs inside its shell. Already it has muscular control. Before hatching, the amnion bursts, but by now the yolk sac is almost absorbed into the chick's body. On the 20th day, the chick breaks out of its shell to become part of the world we all see. Perhaps these pioneering filmmakers have ensured that even the familiar will never seem quite the same again.
Wild Secrets will be back in 2 weeks' time.